So for the past decade, we collectively have been working towards a goal, and that goal is, and we're sharing this belief, that the use of DNA barcodes are going to make it possible to revolutionize biodiversity science. It's going to revolutionize it by making it possible to speed the registration of species, by allowing us for the first time to read biodiversity. There's been a lot of progress over the last decade, but I'll have to say we haven't achieved the revolution. It's a matter of numbers. We've gathered 6 million barcode records over the past decade. That translates into 1 barcode record for every 100 square kilometers on our planet. This is a nearby magnificent nature reserve, the 3rd largest canyon on the planet. It's about 290 square kilometers. There are 3 barcode records in those 300 square kilometers. You don't revolutionize biodiversity science like this. We need monitoring programs that deliver billions of barcode records, ongoing survey programs. This is the way that we'll revolutionize biodiversity. That's deep barcoding. The national parks of our world, this wonderful, the Kruger, the other 18 national parks in South Africa, they surely should be early targets for the assessment of biodiversity and monitoring biodiversity. And the national parks within South Africa are a tiny fraction of those around the world. There are over 3,000 national parks, and we know very little about the organisms that live in them. It's an impossible task today, in the past, to imagine censusing species in all of the national parks in the world. That's an indictment of our discipline. Let's just look at a microcosm. And it's a microcosm that was examined just a few years ago. This was on the cover of Science 5 years ago, almost precise the date. It's a wonderful study. Hebert and colleagues took on the challenge of investigating the invertebrate fauna, the arthropods present in a tiny fragment of one tropical forest, a 0.5 hectare plot in Panama. And it was really a tough challenge they took on, the first spot on our planet. And those red lines, well, how did they do it? Well, they did it with the help of 100 taxonomic colleagues who joined forces to evaluate the specimens, and they did it morphologically. And you can see these large blocks of red. Despite having 100 people involved, there were not taxonomic specialists to work on about half of the groups of arthropods that were collected. The other things that had to be winnowed out were all of the immature individuals. So the census only looked at adult specimens, which were much less, and in these selected groups, there were much less than half the specimens. 130,000 specimens were analyzed, and of those 130,000, 10% of them were identified to a species, and the amount of time required was 1,000 hours, and the estimated cost was $50,000 to do that survey in one site. There's an important lesson from this study. Unless things change, we'll never know the species that live in those national parks. Never. We live on a planet. We've been studying biodiversity for the past 250 years, as you well know. Despite that fact, we believe that no more than 20% of the species on the planet have been registered over that very long interval. Massive numbers of undescribed species. These dark taxa, what will it cost to bring them into the Linnaean system if you're employing morphological approaches? The latest estimate would be about $400 billion to register those 8 million species. That means we'll never know the species on the planet unless things change. And of course, things are changing. We're never going to walk away from the fact that we're in love or fascinated by the analog gradients of life, the variation in size and shape and color. We're never going to lose that. But we're never going to be able to understand life if we live in the analog world. We must move into the digital world, and of course that's what the businesses that we're engaged in is moving biodiversity in that in that digital direction. And there's been substantial progress over the past decade. But the next decade is going to see spectacular advances. Want to look back. We can't shrink that thing a bit because we're losing the bottom of all these slides, can we? Maybe not. Don't worry if it's a giant problem, but it would be nice if we could have everything on the screen. I just want to look back a decade because about 10 years ago there was a meeting in which we brought together that group of people that you can see there. Quite a few of them are in the room today, which is just great. It took us 2 days to decide upon a name for this project, International Barcode of Life. It took us 2 days to decide upon the scientific goal of that project, assemble DNA barcodes for 500,000 species and do it within 5 years. That was the vision. Okay, let's do it. It wasn't that simple. It took us 3 years to raise the money before we could begin the project. So that's an important lesson from the past. It's very easy to come up with big ideas. It's often much more difficult to fund them. But in 2010, September 2010, we activated the International Barcode of Life project formally, and within slightly less than 5 years, we achieved the target coverage of 500,000 species. And one of the problems when you achieve a target is what next? So 2 years ago, at the last meeting, I proposed a biodiversity moonshot. I proposed that we consider activating a mission for planetary biodiversity, and that this mission would have 3 key goals. It would aim to register all of the species on our planet, all of the multicellular species on our planet within 20 years. It would create a library of life, DNA extracts for all of the species that are inventorying during the course of this census. And the other very important thing was to deliver a biosurveillance system for our planet, the capacity to read life on a planetary scale. So those are the 3 goals. And when you're launching a moonshot, it's useful to know a couple of things. One, it's nice to know how far away the moon is. We don't know how far our moon is. We don't know how many species live on our planet. And we also really, well, we did know one thing, and we knew that even if there were 10 million species, it was going to be quite ferociously expensive to carry out the work. We would need billions of dollars to execute this project, and so quite frankly, 2 years ago I was intimidated by that prospect and thought that we needed to do a couple of things. One, we had to gauge the distance to the moon a little bit better, and we had to try and shrink the costs. And so that's where we've been spending the last couple of years. And we've been using the microcosm in which we live, Canada, which covers 7% of the planet, as part of this scoping exercise. It's thought to be home to about 1% of global biodiversity. And so we've asked the question, let's bring barcodes to bear, and are those early estimates of biodiversity in Canada accurate or not, and what can they tell us about planetary biodiversity? So we looked at the vertebrates of Canada, we looked at the plants of Canada, and basically got rather nice concordance with accepted species. There was no major changes there. We then focused on insects, which is the largest compartment of terrestrial biodiversity, and when we looked there, the situation was very, very different. We increased the species count for Canada from about 30,000 species to about 65,000 species in the few sites that we examined. And of course, Canada's a big place. We didn't sample them all, and when we put the richness that we had encountered, the estimate for Canada jumped to 94,000 species, nearly trebling the species count for our country, and having an impact on the estimated number of insect species on the planet if the 1% rule is applied. The latest estimates were about 5 million species, 5.8 million species of insects just a couple of years ago. This drives the number up to about 10 million insect species. The moon is getting further away. And of course, it's not just insects that are thought to be hyper-diverse on our planet. There are a number of invertebrate groups in which we believe there's lots of diversity, but we don't have any real idea of how big they might be. The mites of the planet, the harpacticoid copepods that are dominant in marine environments, and of course, nematodes that are thought to be ferociously diverse. So we've begun assessing the diversity in one of these compartments, And these are the results for Canada, the accumulation curve, relatively small sample size, only about 70,000 specimens run, and about 10,000 species, slightly less than 10,000 species encountered, and the projected curve, about 22,000 species of mites in Canada. And when you turn that into a global estimate, that adds 2 million species to the species count for arthropods. And if it so happens that harpacticoids show the same numbers, which is certainly possible, and you add in the nematodes and the other animal phyla, and you add in plants, and you add in fungi, Pedro, I think you just said to me, millions of species of fungi, it's almost certain we're over 20 million species of multicellular organisms. The distance to the moon just doubled. That's what the dark taxa grid looks like now. The known taxa just shrank. We've got a lot of work to do to register species. What about the other large part of the planetary biodiversity mission? This view of developing a system where we're reading, censusing life across the planet. What are the factors that complicate that? There are two factors. One is the number of species that we've just talked a little bit about. The other is the amount of geographic patterning in life. If life was the same everywhere, the task would be easy. If it's very spatially structured, it's going to be a more complex enterprise. And of course, when you look at what we know about biogeography, we know we've got a couple of hundred years of censusing life on the planet. We know about these large biogeographic regions. We know more about biodiversity patterning on land than we do in water. So it might make sense to begin with terrestrial organisms. And of course we know there are more species on land than in the oceans, so that's a bad thing. But on the other hand, if we can conquer biosurveillance in a terrestrial environment, we can spin this out into fresh waters, we can spin it out into marine environments. And so the approach that we've taken is tackle the most diverse group on the planet and try and develop a biosurveillance system for it. These are the ecoregions of the world. There are 867 ecoregions of the world. There's a lot of spatial heterogeneity in life on our planet. Wouldn't it be nice if we could just drop into each one of those ecoregions, sample it in just one place, and that would give us a reading of biodiversity for that ecoregion? It's not going to be that simple. We began by looking at national parks in Canada. We have 45 of them, and assessing their arthropod diversity and asking how similar they were and doing every pairwise combination of those 45 parks and looking at the similarity in arthropod communities. And here you can see a relationship which is no surprise, and a curvilinear regression. Distance between sites here and the similarity in the fauna and this decline. Distance explains 37% of the variation in arthropod communities in Canada's national parks. Of course, the question is, what about the residuals? What explains all the rest of the variation? And that then led us to think about the ecoregions in Canada. And as I mentioned, we have 15 of them. And I don't have time to talk about all of them, but I'll talk about the biggest one, the boreal shield, and I'll compare it with the montane cordillera. And the simplest thing you could ask is, are geographic patterns similar in all ecoregions? Is this big one got more heterogeneity? The montane one? And the answer is absolutely not. These are the results, distance along here, and the similarity in community in that giant boreal ecozone, and you can see there's almost no change in the insect community. You can sample from Newfoundland out to Alberta, and the insect communities are almost identical. Go into the montane cordillera, and there's very rapid changes. You need to do more sampling in the montane cordillera if you want to understand life in Canada than you do in the boreal ecozone. So this kind of information is providing, I would say, some useful insights on how we might set up a biosurveillance program in Canada. But you can't use this to model the world. And that's why we launched a global program a few years ago to survey arthropod diversity patterns at sites across our planet. We've now deployed these particular traps that you can see here, a Malaise trap. It's a beautiful passive sampling system. We wanted to be able to compare sites across the planet. We deployed the traps in cities, downtown Washington, Paris. We deployed them in Johannesburg, We deployed them in nature reserves, protected areas across the planet. So this is a very light, site-based sampling, and we ask what kinds of patterns do you encounter? And this is the first signal you get. 1 million specimens analyzed, 120,000 nearly insect species encountered, and gee whiz, it doesn't look like we're anywhere close to finishing the job. In fact, my expectation is we may need to analyze 100 million insect specimens around the world in order to hit an asymptote. That's one big group of life, but it's still 100 million sequences. What else did we learn? Well, one of the surprises, I certainly went into this preconceived idea that if you put traps along a latitudinal gradient, you're going to see very few species in the Arctic, you're going to see more in the temperate zone, and you're just going to see a lot more in the tropics. There's Greenland, way down there with the lowest count. Okay, that's nice. Here are the temperate zone sites, and here are the tropical sites. Yeah, there's a trend line. Latitude explains 36% of the variation in insect diversity, and many temperate zone sites have as much diversity or nearly the same diversity as tropical sites. There's only 50% more species in the tropics. No, that's not right. What's going on? Whoops, let's go forward. This is what's going on. Think back to Canada. This is distance along here. There's the boreal shield. Very low turnover, as we said. More in the montane Cordillera. This is Costa Rica. In 30 kilometers, you see tremendous turnover in the insect community. We're going to have to do intense surveys in tropical regions in order to assess insect communities. It isn't true that tropical insect communities are only 50% more diverse than those in Canada. There's just a lot more spatial heterogeneity. Okay, I said we wanted to carry out a scoping exercise for planetary biodiversity. Register all species. What have we learned? Well, we doubled the species count. The estimated cost, $3 billion. Library of Life is going to be pretty cheap because we get the DNA extracts as we carry out the work, maybe $100 million. And this project, global biomonitoring, is going to be ferociously expensive unless things change. So I'm afraid our work didn't much less intimidate me, but it did really stimulate us to look for cheap ways of doing this. So that's where I'm going to shift now. How can we launch PBM in an affordable fashion? Well, of course, we're going to make use of new sequencing technologies, and there are two main sequencing platform types. The short read platforms, they really struggle to deliver 500 base pairs of sequence information. They've got error rates of 1% to 2%, but they can generate up to 300 million reads in a run. And then we have long read platforms, the third generation. Don't do that again, Paul. Whew, didn't explode at least. Good way to get me off the stage, Michelle. Long read, which can generate reads up to 50,000 base pairs. So these are the solution. Just before we jump into the solution, let's just remind ourselves that we've got two analytical paths that we're following in barcode research. One is barcoding and the other is metabarcoding. And metabarcoding, of course, is the analysis of bulk samples. And what it's really super at is species occurrence data. And it's very inexpensive. It's probably 100-fold less expensive than specimen-based analysis. With specimen-based analysis, barcoding, you get a lot more. You get the reference library, you get specimen identification, species discovery, species occurrence, species abundance. There is a reason why you might want to keep doing reference library construction, because you get a lot of extra added value for it. So let's think about those sequencers. Is there one sequencer that does all? And the answer is no. These short read platforms are excellent for metabarcoding. You're going to have to crimp a bit on the length of the amplicon, but they deliver lots of reads, and that's what you need. And in terms of barcoding, the SQL is a fantastic platform to deliver barcodes. And you may say, that's a pretty strange thing to say. Barcodes are short. Why the heck would you want to read 50,000 base pairs? And the reason you want to read a large number of base pairs is because the error rates in these third-generation sequencers are fantastically high, 10 to 20% at a particular position. But look at the nice trick. So this is a sequel machine. This is the little smart cell, which is the standard sort of analytical unit on this platform, and you take your CO1 amplicon, or you take your ITS amplicon, and you turn it into what they call a smart bell, and basically it's circularized now, and all of those 50,000 base pairs or 20,000 base pairs of read, just read around in a loop and deliver a consensus sequence. So you average out those errors and get very accurate sequences. Here's the results. Black is Sanger, blue is Sequel. You can see they're effectively identical. Sanger's been the gold standard for reference library building. Sequel delivers sequences that are identical if you don't look too carefully. If you look really carefully, they're better than Sanger because every one of the sequences is exactly the amplicon length, 658 base pairs. You always get truncation with Sanger sequences, even if you do bidirectional reads. There's another rather marvelous thing about Sequel, and that is it doesn't just do a few specimens at a time. One of those little cells that you just saw, which is about the size of, tiny, this plate shows the read count when you load 10,000 amplicons from 10,000 specimens. Make it into basically an amplicon soup, 10,000 different specimens. And how do you recognize that little well? Because every one, you use 100 pairwise combination of 100 forward primers and 100 reverse primers, each of which is specifically tagged. And you can pull out 10,000 different organisms in a single run. And the cool thing, that single instrument, because of that, will run 10 million specimens a year, and the cost to sequence each specimen will be 20 cents, and the sequence quality is higher than we will get with Sanger. So from my view, this is the platform that's going to replace Sanger in large-scale operations, and it is going to have a tremendous impact on our capacity to build a library of life. On the metabarcoding front, any of the second-generation platforms, be they Illumina or Ion, are quite effective. S5, HiSeq 2500 is cool because it generates 300 million reads at a time. And the workflow I think you're all familiar with, bulk of organisms, DNA extract, PCR, generally less than 500 base pairs, so you're not going to be getting full-length barcodes, sequence, bin assignment, magnificent technology, wonderful way to think about biosurveillance. And note we're now talking about hundreds of millions, even billions of reads in a particular study, and this then creates a bioinformatics hurdle. And we've been working over the last couple of years, the informatics team at the CBG, our Center for Biodiversity Genomics, has been working to create a new platform that really makes it very easy to analyze these large data, and Sajeevan will be talking about it on Friday. I don't have time to do more now except to say there will be an informatics platform, a public informatics platform available to support the analysis of these, to tame the billions of barcode sequences. So earlier this year, we were sitting, okay, we've got the sequencers, we've got the analytical platform, we've got to do something that will be fun. Let us analyze 1 million specimens in a week. Why not? So we have lots of specimens sitting in freezers because we've never been able to afford to analyze them before, so we decided to focus this project on 450 samples that were collected from provincial parks in Ontario, Malaise trap samples again, and we took those 45 sites and split them into 2 groups. Half of them we did just as metabarcode, and the other half we did as both reference on the Sequel and as bulk samples. So this is the kind of thing you're doing. On the 256,000 specimens, we chased 658. There's the specimens, each one vouchered. On the S5, we're doing bulk samples, 750,000 specimens, chasing a slightly shorter amplicon. How long does it take to run those 260,000 specimens? Put 9,000 in each smart cell. You can run 4 smart cells a day, 1 week to run 260,000 specimens. And when you do that, what do you get? Here's the arthropod diversity curve for those provincial parks. 14,000 species encountered. Only 11% of them were new, which was really great. That meant all of our prior work in Canada has registered almost all of the insect species. And you'll note that only 5% belong to the new bins, so the new bins are rare things. But we still encountered 1,200 new bins, so I'm sort of happy we did some reference library expansion. And we've done a lot of Sanger analysis, and here you can see Sanger results for Ontario. Here you can see sequel results from Ontario. Very similar. The sequel's doing maybe a little bit better in recovering taxa, but the cool thing, look at the cost difference. $36,000 versus $1.2 million. So reference library building just became very feasible. So what about the bulk samples? We ran them on an S5 platform because we didn't have that many samples that needed to get run. 12 bulk samples, 4 chips a day, 750,000 specimens, 228 million reads. That's 1 week of machine time. There's the species accumulation curve.
Good morning.
Nearly 20,000 species were encountered in this larger sample. So let's glue these two together, and let's try and use the forward as opposed to the light. Works better. And there's the combined. We sampled nearly 25,000 species in the course of 1 week of machine time. And one of the things about this, think back to the study in Panama and all the red. All of these species, all of these taxa were analyzed. Nothing was excluded. Everything obtained a species level designation.
Shall I just do that then?
The technical skills required to do this are very low. This is our senior lab technician. He works 2 hours a week. You know, it's ridiculous.
Yes. Yeah.
So what have we done in Ontario in the course of this project? Here is the distribution of parks and the bins. And one thing you can see is nearly a third of those 25,000 taxa showed up in only one park.
Yeah.
There's more species to be found in the Ontario parks, but the other thing we got for every one of these parks is a long species list.
Cool.
There were no species lists. From 1,200 to 4,500 species we now recorded in those parks.
Yeah.
And this diagram shows the connectedness for just 17 of these parks.
Yeah.
The block in the center is the thing was only collected in that park. And these are the linkages. This chord diagram shows the linkages. The species overlap with every one of those other 16 parks. You don't want to look at the 45 park chord diagram.
I'll close it to that.
This is the most extensive data set that we have anywhere on the planet for arthropod communities. Who cares? Earlier this year, there was a paper in Science that asked, where have all the insects gone? The data was this. Malaise traps deployed in Germany showed an 80% decline. Biomass in 1989, biomass in 2013. 80% decline in insect populations. Subsequent papers have reaffirmed the generality of this observation in other sites in Europe and in North America. Connected to it, declines in bird population like the shrike. From this nature reserve and all of our insectivorous birds in Canada are under major decline, 75% decline. All of these studies share a weakness. The only information we have is biomass. Don't you think it might be useful if we actually knew what collapsed? Wouldn't it be good to know if big insects collapse like little insects? Wouldn't it be good to know if all insect orders, or if there were very selective biases? It's past the time when we should be treating a major group of life on our planet as a black box.
But I've got those guys.
This is an indictment of our discipline.
Yes, I'll add you to the group.
So we've been thinking about how you might carry out biosurveillance on the planet. And we began by fracturing the planet into 4,000 blocks, each 150,000 square kilometers. And then we fracture each block into grids like this, 2,500 kilometer square grid, 50 kilometer by 50 kilometer.
All right, man.
And we view these as sampling stations, and the idea of injecting a sampling system somewhere in the middle of each of those grids. So moving from site sampling to grid sampling, South Africa has 7 blocks. It will take 420 sampling sites to survey South Africa intensively. I wish I lived in South Africa, because in Canada we have 70 blocks. It will require 4,200 sampling sites to sample Canada. So we're looking for a way out, and the way out is to return to our ecozones and say we're going to focus our grid sampling on an ecosystem by ecosystem basis. And we're beginning next year with this one down here, and the southern part of Canada closest to where we are. Whoa, damn. And this is implementation. There's a sampling grid. Imagine a sampling system right in the middle of those. It'll be Malaise traps in the future. It will be more. We had to add not just 60, but 82 because those damn lakes got in the way. And we want to cover 150,000 square kilometers. We want to cover 1,000th of our planet's surface. So we'll be sampling next year. And what will it cost? We're expecting to collect 2.5 million specimens. We're going to gather 10 million sequences per site. And the cost for this whole program, sampling will be $44,000. Sequencing will be $74,000. It's about $100,000 for analytics and about $40,000 to carry out the sampling. And of course, we're not planning to just do that one grid. We're going to work our way across Canada and survey 7 of the major ecoregions over the next few years. And we're working towards this in 2023. The activation of a biosurveillance system for the planet. We will sustain grid sampling permanently. We will supplement it with targeted sampling in our national parks, and we'll supplement it with targeted sampling around all of our major ports so that we can intercept invasive species. We will also have stations at all of the agricultural research facilities in Canada. This will be a biosurveillance system for our nation, and it will cost about $1 million a year to execute. And you may say, well, I'm not that interested in arthropods. Maybe we should be thinking about other things. But I'd like to, and we expect to not just be looking at arthropods, and we really think Malaise trap sampling is really biome sampling, because insects, these little insects, every blood meal, every bit of blood they suck out of a vertebrate, every bit of pollen, every time as they go about their life, they're picking up DNA from the environment, bringing it back to that bottle. You pick the right primer sets and the right gene targets, and all of a sudden you're reading the biome, not just bugs. So this is our plan for the biosurveillance, biome sequencing, biome analysis. And some of you may be wondering, well, you know, what about the aquatic environments in Canada? What about the marine environments in Canada? We think you need role models. We think you need to show that you can do something, and that will be the attractant. That will cause and I don't believe the rest of the world will be in diapause. I'm convinced that it will pick, places around the planet are picking up and doing this as well. Let's look back to the 19th century. And what are we looking at here? These are weather stations in North America, and the seasonal flux in temperature through each year. And you see 1940, what happened? There were just a few stations in 1880, and 50 years later, our continent and the world, in fact, was awash in weather stations. And here you can see now in this decade, some new stations, some new gaps filled. This is what happens when you establish monitoring stations that deliver information that's valuable to humanity. You don't turn them off. And I'm sure that the weather station analogy is the right one for biosurveillance. So now, let's think about planetary biodiversity. I said that 2 years ago I was intimidated. I was intimidated to hell. I'm not any longer. I'm sure that we can census the 20 million species on our planet for about $300 million. I'm sure that we can preserve the DNA extracts for another $100 million. And I think we can set up a global surveillance system that's looking at 5,000 sites for perhaps as little as $20 million a year. These are inconsequential sums in the world of big science. This project is now something that we can afford to do And collectively, I'm convinced that we should work towards its activation in just the next few years. So let's think about a world over the next 20 years, a world in which it's not just Canada, but it's the Kruger that's sampling its biodiversity in this ecoregion. And slowly but surely, our planet goes green. And slowly but surely, the weather system, the biodiversity monitoring system is put in place around our planet. And our world is changed. And all it took was some deep barcoding. I like watching this thing spin, but it will spin to your part of the world and end up there, because this is a very important part of the world for biodiversity monitoring. And I really hope that by 2035, this is the way our planet looks. In 2010, I think an outsider might have said, biodiversity science is broken. You don't know how many species are on the planet. You've never managed to survey a single site comprehensively. In 25 years, I'm pretty confident we'll say, we completed the inventory of life, and we have a biosurveillance system, and we're protecting that life. And all it took was a bit of deep barcoding. Thank you.
Hey, Paul. Fabulous talk. Insightful, as always. You're referring to bins when you're referring to biodiversity rather than identified species, correct?
Correct.
So what do we do about the massive number of species which have not and perhaps will not be identified taxonomically for long periods of time? Is that important?
Well, I mean, I think we're all very interested in seeing, a completely dark bin. That's where the microbial people live. I don't want to live in that world. I want to understand as many biological covariates of these organisms. So if today, Hugh, you were to go out and collect just about any animal in a terrestrial environment anywhere on the planet, it would be assigned to a bin, and there's a very high prospect that that bin is going to coincide with a taxonomic species. So that's good. The other thing that we can do is we can position that bin with no more information than just that short sequence to a particular family of organisms on the planet. So it's not completely dark. I am sure that many of these species are going to take a very long time to be described. The sesiomyids of the world, where there may be 2 million or more species, very long time. As long as you have that sequence cluster, 20 years from now, when that's described, that specimen gets a retrospective identification, right? So I think the big job for us is to rush forward, register life on the planet, create a lot of dark bins, and I think that will be a motivation for humanity to want to add biological collaterals to it. Is that? Yeah. Thanks. Thank you, Michelle. Thank you.